Almost every tumor, colorectal cancer tumor, is mutated in APC. In both copies are mutant. Those mutations tend to be mutations in the DNA sequence that lead to a truncation. So there's a stop codon, so the protein is truncated. So there's only bits, the N-terminal fragments are very loosely expressed. And it turns out when that's the case, cells undergo all kinds of changes. A really important one is that there's this whole Wnt signaling pathway that is then. There's a downstream target of beta-catenin that is stabilized when APC is not there, and that leads to changes that keep cells proliferating and stop them from differentiating. That's one thing, or they don't differentiate the same. That's one thing. And in that context, some of these other functions of APC, it binds to microtubules, it stabilizes them. That is important for normal mitotic spindles, that's important for migration, that's important for cytoskeletal stability. So you have cells that now proliferate more, you have cells that don't migrate quite as well, they don't divide as properly, no, they make more mistakes during division, and turns out they also don't die when they should. So now you have the perfect foundation for building a tumor. The cells divide for longer, they make more mistakes, the mistakes are tolerated, and the cells are not eliminated. So in the environment of the gut, that's actually, it's like a... So I focus on APC because it's mutant in almost every colorectal cancer cell. So I think if we understand what is it that losing or mutating that turns it into a tumor cell, that gives us an opportunity to say, well, what are the changes that have happened and how can we target them to either reverse them or eliminate those cells specifically? We know that because of this mutation, all these things go wrong. We can say, well, that means that cell is now particularly sensitive to this thing, then we could potentially eliminate them selectively. So that's one thing. When a cell divides, it builds a mitotic spindle. But it also, and the mitotic spindle is made up of microtubules, and it's a structure that kind of looks like this. And in between, my fingers crossed, is where all the DNA is lined up and the chromosomes are. And then at some point, when the cell is satisfied that everything's attached properly, everything's been replicated, everything's been aligned, each microtubule end on each side of the spindle, it touches only to one of the sister chromatids and the other does the other, then there's a checkpoint that monitors all those things. And that is satisfied, the whole thing of a string of event happens that allows the separation where the mitotic spindle actually acts and pulls these things apart to build two new daughter cells that supposedly have the same genetic material. And that checkpoint is really important because if it doesn't act and there's the thing aren't quite right, you can get two chromosomes into one cell, or it could break it, or things can go wrong. So normal cells have this checkpoint. But if you add a microtubule poison, that spindle isn't normal. Either a microtubule poisons come in two flavors. One is microtubules are completely disassembled, so they can never build a spindle. The other one is they can't disassemble, and you need dynamic turnover of assembly and disassembly to get the spindle to build. So in either case, you have abnormal spindle. In one case, you have no attachment, and this checkpoint that I mentioned that allows the cells to progress depends on normal attachment and also on tension on the chromatids. And when that's not there, the checkpoint should say, stop, 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 I'm not ready to go, I'm not ready to go, I'm not ready to go. And that causes a prolonged arrest. So these poisons cause a prolonged mitotic arrest. And that should actually tell the cell after a certain time, something is so wrong, I need to die because I can't divide normally. And so when that connection isn't right, either the cell doesn't register there's a problem in mitosis, or it registers but it doesn't execute the appropriate exit, then either of those things are problems. And so not having APC, we know, compromises the former, i.e. the checkpoint doesn't report quite right. And whether it also does the latter, we don't, that's really where we're trying to go. So what this paper showed were really two important things. So because we knew of this connection between APC and microtubules, and because all colorectal tumor cells have mutant APC, well, all, for intensive purposes, all, and so we thought, well, that should make the cells more sensitive to these poisons. And microtubule poisons are already used clinically. So you'll have heard of paclitaxel. which is used in breast cancer, for instance. And again, you might think I'm diverging, but I just want to set the stage. And these microtubule poisons are used clinically because cancer cells, obviously, we have the idea that they divide more and for longer. So they should be particularly sensitive to something that creates defective divisions and creates an arrest, because they rely on this more. That's the idea, and so that's why they're used. So we thought, well, gee, if all colorectal cancer cells are mutant in APC, then and we know they have problems in microtubules, because APC is one of the functions, are they more sensitive to these drugs? So we tested a whole bunch of them. And much to our surprise, initially we found that actually they're more resistant. Now, and then we discovered that has to do with this checkpoint being defective, and so they don't care about arresting. They don't arrest. They just go on their merry way and make mistakes. So that's one thing. But then we tested some other ones, and then we found this one particular drug, vinorelbin, which is a vinca alkaloid. They were actually more sensitive, surprisingly. And if you look at the first figure, that's what we show. So that was, we said, well, that's interesting, but why is that for cortisol and Taxol and some of these others? These cells are less sensitive. So what's the difference between the two? And then we discovered that, well, vinorelbin actually, unlike what we had thought, doesn't kill cells just because they've arrested in mitosis for too long. It actually kills cells regardless of the cell cycle they're in. So all the other drugs, we always think, oh, what they do is they arrest cells in mitosis, they are arrested too long because there's something wrong, and off they go and die. Well, we knew that didn't work for APC, but vinorelbin doesn't do that. It kills by a different mechanism, and we don't really know how, but because it doesn't require the cell to arrest in mitosis. It's just if a cell is an interface and you give it this drug, it doesn't then go into mitosis and then die, it just dies. So using vinorelbin is potentially a good therapeutic for this disease. Lots of horrible side effects. It hasn't been used in colon cancer, and I don't know, it hasn't really been shown that effective in mouse. It works quite well. So we do show the data that when we treat mice that are prone to get cancer with vinorelbin, we do get more cells are dying and the tumors are smaller. The problem with all of these things is they're also toxic because lots of cells will have response. And so whether it's something, I think it's important to say, look, this could be really useful, but because of the toxicity and often also the distribution into the body and things like that, it's not always as clear cut because the tumor doesn't always get all the drug and goes many other places. And what this shows is, though, that we need to think about these drugs differently. They don't exactly work the way everybody thought they worked. And what cancer therapy is beginning to realize is that just giving one drug will never be enough, because tumors are heterogeneous and they can develop resistance. And so because they're heterogeneous, some cells will be killed very well by these drugs, but there will be others that have another set of maybe overlapping but slightly distinct mutations that makes them more resistant. And those, it's just like when you treat an infection, when you have an infection in your throat or wherever, then those can become resistant very rapidly, because that's what you select for if you don't give a whole ton of antibiotics. So, and in infectious diseases and parasitic diseases, for instance, in tropical diseases, you wouldn't be allowed to give just one compound. But in cancer, that's what we still do. And in colon cancer, this is the one thing I need to really say is we still don't have really good drugs for it. The mainstay chemotherapy or therapy for colon cancer is radiation therapy and 5-FU, which is a DNA damaging agent that's been around for a long, long time. And it works for some people and it doesn't work for others. Cocktail. So maybe we should look at the tumor spectrum and we say, well, this patient has this and this mutation, well, it's got this APC mutation, so we know that's maybe. But we also, for instance, I'm just writing a grant so we can be much more, to put all this information together and to understand, well, when we hit with vinorelbin, which cells actually die? Is it the ones is it just the ones that are stem cell? Well, that'd be kind of cool. Or is it other ones? We need to understand that much better. So we can say, well, we really need to hit these. We need to hit the stem cells, the non-stem cells, the secretory cells, and they need to all be targeted, and they may require different things. And the nice thing about vinorelbine is because it doesn't require mitosis, it could also kill cells that are not dividing. And so that's really exciting. But there are many cells that are not dividing in tumors, and that may be why we see them, because the tumors get smaller because it's targeting those cells. We need to put that on our list of things to try or to include, depending on what a tumor is like. Is it the only one? No. And because there are too many other factors at play. But it adds a piece of information that I think is helpful that we didn't have before.